IVM.
Dear listeners, thank you for tuning in to the Pragati Podcast every week. This week, we are rebroadcasting an episode from September 2018. Genome sequencing and mapping is unearthing new knowledge about the origin of humans and human civilization. More than that, genome analysis is also yielding new insights about human health and will be vital for the medicines of the 21st century. Since this episode was first aired in September 2018, there's been fascinating new research expanding our understanding of how humans populated India and South Asia. Most recently, the first ancient genome from the Indus Valley Civilization was analysed and the results were published by a large group of researchers, including our guest today, Dr. Thangaraj. This episode takes a brief look at history and focuses on what new genomic information means for the health of all Indians. We'll be back with a new episode next week. Stay tuned.
Welcome to the Pragati Podcast, a weekly talk show on public policy, economics, and international relations. We are your hosts, Hamsini Hariharan
and Pavan Srinath. Joining me as a guest host today is Dr. Shambhavi Naik, fellow at the Takshashila Institution. Dr. Thangaraj is a renowned scientist working at the Center for Cellular and Molecular Biology, CCMB, in Hyderabad. He has published extensively on the origin of humans, on genetics and disease and how they affect the lives of Indians. He has a host of publications in top journals like Nature and Science and is at the cutting edge of his field, not just in India, but in the world. We'll be back with Drs. Thangaraj and Naik after this short break.
Hello and welcome to a brand new week on the IVM network. If you're not following us on social media, please do. We are at IVM Podcast on Twitter, Facebook and Instagram. If you're listening to a show of ours and you like it, please take a screenshot, tag us. We will reshare, repost and retweet your screenshots from our account. Here's what's in store for you this week. On Cyrus Says, Cyrus is joined by Sarang Sate and Paula McGlynn of Bhadipa. Tune in for a fun conversation about how the Marathi digital platform came about. This week, IVM Podcast's co-founder Kavita Rajwade and Karthik Nagarajan take you through the journey of the Filter Coffee podcast. They share their favorite memories from renowned guests and talk about what to look forward to in the show's future. On Marbles Lost and Found, Zain and Avanti host an AMA special where they answer listener questions. On Mr. and Mrs. Binge Watch, Janice and Anirudh continue their Emmy special series. Tune in to know their picks from the comedy category. On Water Player, Mikhail, Akash and Siddharth are discussing the Ashes, the Afghanistan-Bangladesh match and a lot more. On Vartalab, it's a crossover episode. Hosts Akash and Naveen are joined by Anupam Gupta, host of Paisa Vaisa. Tune in to discover a different side of Anupam on this podcast. On Agla Station Adulthood, Ayushi and Ritasha discuss how to navigate through relationships as adults. On Pulyabazi, host Pranay talks to Samrat, a writer and journalist, about the causes and dynamics of the tensions in Northeast India. On Kinetic Living, Coach Urmi shares a workout called X Cross MC on Tabata Tuesdays and also highlights the importance of resting between workout routines on Thriving Thursdays. On Geek Fruit, hosts Tejas and Dinkar turn the tables from last week and this week discuss singers who decided to be actors and how they have fared. On the Pragati Podcast, Manoj Keval Ramani joins our host Pavan to explain the ongoing Hong Kong protests and its history so far. And with that, let's get you back to your show.
Welcome back. Hi, Dr. Thangaraj. It's a pleasure to have you on the Pragati Podcast.
Hello. Yeah, thank you very much for inviting me to speak here.
And Shambhavi, welcome back. So, Dr. Thangaraj, you just today morning gave an excellent talk at Takshashila about your work on the history of humanity as unfolded by genetics, not just by archaeology and anthropology and other disciplines. We are in an age where genetics is able to give us more information and advance our understanding of history even before writing and so on. And we also find that the same history is also linked to our current human health and disease. So, if I can ask the first question, how long ago did humans come to India and how did we find out about it?
So the modern human originated in Africa about 160 to 200,000 years ago. And they started migrating out of Africa. Our work on Andamanis actually defined that the modern human must have arrived Indian subcontinent around 65 to 70,000 years ago.
Does this make the Andamanese population one of the oldest discovered outside Africa?
Yes. After that, people have also studied Australian Aboriginals.
Okay.
But they are not pure. They are all admixed. The pure population who migrated out of Africa are the Andamanese.
And that's because of the quirk of geography, right? Andamanese have sort of remained as an isolated community.
They remained in isolation for last several thousands of years. So that makes them as one of the very unique populations, very unmixed, unique populations. Still they carry their own genome.
Could you tell us a little bit about the science of how you can tell that they came to Andamans about 70,000 years ago?
So we used to use several type of markers. One is the markers which are inherited maternally. That's the mitochondrial DNA marker.
Okay.
Other one is inherited paternally. That's a Y chromosome. And there are also markers which inherits by both the parents. It's autosomal markers. So using the uniparentally inherited markers like mitochondrial markers and Y chromosome markers, one can trace back the maternal and paternal lineages. By also looking at the mutations in the mitochondrial DNA, one can calculate the age of the population.
Okay.
Since the Andamanese are in isolation for the last several thousands of years, they acquired unique set of mutations. So those mutations have helped us in calculating the age of the populations. That turned out to be 65,000 to 70,000 years.
The mitochondrial DNA are little circular pieces of DNA, right? They're not there in the nucleus. So they don't come from both the mother and the father, right?
Yeah, in a cell, we have nuclear genome. In a somatic cell, that means any part of the body, if you take a cell, it consists of nucleus. That means within the nucleus, there are 46 chromosomes.
Right.
Of that, 23 we inherit from the mother, 23 we inherit from the father. Whereas the mitochondria, in a given cell, there will be large number of mitochondria, and each mitochondria there will be a large number of mitochondrial DNA. They are all inherited only through the mother, because only the egg mitochondria remains there. The sperm mitochondria remains outside at the time of fertilization. So there is no paternal contribution of mitochondria.
So that way we can trace it back generation on generation.
Yes.
Without this cross-fertilization.
Yes,
right?
yes.
Okay.
So, Dr. Thangaraj, if I may just ask, what are different sources of mutations that actually contribute to how the genome looks? And why is population isolation such an important factor in this case?
If you take mitochondria, different region has a different mutation rate. Mitochondrial DNA is 16.5 kilobase in length, circular DNA. Unlike the nuclear genome, there is no intron. All the genes are arranged one after the other. On the other hand, there's also a region called hypervariable region. It's about 1.5 kb in size. So only that part is non-coding. Mm-hmm. Therefore, the mutation rate is very high within that hypervariable region. So that helps if somebody wanted to look at immediate individual variations or population-specific variation. One can use that hypervariable region to find out this difference. But somebody want to look for the evolutionarily conserved variations, they can be found only in the coding regions of the mitochondrial DNA. So those are the ones which we use to trace back the evolution of different populations. They are very, very consistent in nature.
So you have mutations that happen in certain regions, once every few generations, once every few 100 years?
Yeah. So there are mutations. For example, the hypervariable region is the one which has a very fast evolving mutation rate. Maybe once in 400 to 500 years, one can find the mutation. Whereas in the coding region, some region can have mutation once in several hundreds or several thousands of years.
Okay.
So that helps at in various level or various evolutionary stage, one can calculate the age of the population.
Okay, so in that sense, just like we have seen all these species trees, right? Evolutionary trees of various species. So what you're telling us that even within human beings, we can find something like an evolutionary tree of populations by looking at something like the mitochondrial DNA.
Yes. So one can, there's a scientific term called as haplogroups.
Okay.
So based on specific mutation, the population or people can be grouped in a specific haplogroup.
Okay.
So that is based on the mutations
right
in a mitochondrial DNA. For example, the two populations are very recently diverged. You can see the most of the mutation would be common between this group. There will be very few makes a difference. So as I mentioned earlier, that different set of mutation will have different age. So one can actually calculate how many hundreds or how many thousand years back these two populations have diverged.
Okay.
So that's the advantage of using.
And similarly, you can also find out about the male line via the Y chromosome.
Yes.
Okay.
Yes. So same way the Y chromosome also can be used for constructing the haplogroups. So based on the different set of mutations.
So we know that the Andamanis are among the oldest human beings with a unique population discovered outside Africa. But who are we as Indians today then? Where did we come from? I know it's a grand question. There are all kinds of debates about Aryan invasion, migration. But what is the base of what genetics is telling us?
Yeah, it's very interesting for everybody to know about our own origin. So we all part of, particularly those who are in South India, are part of the early human migration. Actually, when the Andamanis were migrating, taking the southern coastal route, a group of them have stayed back in southern part of India.
Okay.
Some migrated to Andaman Islands, some stayed there, some migrated further to Australia.
Okay.
If you look at Australian Aboriginals, Andamanis, and some populations, South India particularly, population like those who are in Kerala, Kurumba, they all share a common genetic affinities. So that tells that this is the way they have all migrated. In this process of studying human population genetics, we came to know that the first modern human who settled in India are the one who migrated earlier as a part of Andamanis migration, who settled in southern part of India. We call them as ancestral South Indians.
Okay.
That is the first settlement in Indian subcontinent. Then the second wave of migration brought people towards north, who are also related to Middle Easterner and Europeans, whom we call as ancestral North Indians.
Okay.
And these two groups have gave rise to many populations, and this had happened for about several thousands of years.
Okay.
Then during the last 2,000 to 4,000 years back, these two founding groups have admixed.
Okay.
This
admixed means intermarried.
They married. There was a marriage pattern between ancestral South Indian and ancestral North Indians.
Okay.
This had happened for 2,000 years. After that, for the last 2,000 years, every single population started maintaining the endogamy marriage. Endogamy in the sense, marrying within the community or within the group. And because of that, so every single population is very unique now.
Okay.
And we don't know what was the reason why everybody started maintaining the endogamy or marrying within the population. One reason could be the caste system which arose around 1,000 to 1,500 years must have put more pressure for every population to maintain the endogamy.
Right.
But the marriage pattern marrying within the group is much older than the caste system.
Any examples of that where we don't have caste but we still have this idea of marrying within the community?
Yes, there are many issues, particularly if you go to the remote place where the ancestral tribal groups are there, particularly if you take even Andamanis. There are 4 tribal groups, Andamanis, who typically look like Africans, like Onge, Great Andamanis, Jarvas and Sentinelese. Although they are all typically look like Africans, but they marry within the groups. They don't marry between the groups. That's the best example. Who doesn't know anything about caste system? No, they still marry, follow the population-specific marriage.
And does this make India unique in the world? Is this? Do you see this level of endogamy that exists in other parts of the world?
You don't see such kind of endogamy and such a large number of population groups anywhere in the world. It has been well established now based on the anthropological evidence. We have 4,635 population groups in India.
Wow.
Nowhere in the world you have such a huge human diversity. This diversity, in a way, very unique in terms of population as a whole, and also the disease pattern is very unique when compared to other countries or other continent. That's mainly because of the population-specific marriage or the endogamy practice for the last 2,000 years.
So comparatively, we'd say Europe, which might be of a similar size, might have only a few hundred such population groups?
Yes, very few hundred, yes.
Okay, so which means that there are specific genetic problems within these communities, and therefore studying India as a whole will not mean much until we study each of these.
Yes, if somebody studies 1 or 2 population and says that we have studied Indian population, so that is not the right, because we need to study every single population to understand the population history, particularly to understand the disease or health aspects of these populations.
It strikes me that apart from endogamy, even till very recently, I think the pre-scientific notion was that the lineage is patrilineal, right? So from your father's line. So even I think within endogamy, there was this idea of gotras within the caste system where you're supposed to marry outside the patrilineal line. But that would something like that have helped maintain some genetic diversity within a community? Or
if you consider in a long term, thousands of years, several generations, that may not really help, because maybe few generations it may help. Again, it's going a circle.
Right.
One has to find partner from the same community. So that's what we are talking about.
Right.
And they're not going to marry anyone from outside that community. So it's making a circle. Maybe it delays a little bit, but ultimately all within the community.
And also just to add, I think with the Gotra thing, you guys still have marriages within the family, because so my aunt, when she gets married to somebody else, will have a different Gotra. So
Right.
I might still be able to marry my cousin. So the DNA shared is still going to be there. Even the Gotra is different, I'm still marrying
and we've
within
heard
family.
of cousin marriage within 2 generations of us in many communities, right?
Yes.
And so what kind of problems can this cause?
Yeah, so that's a very, very interesting study which we did very recently to understand what would be the impact of this endogamy which is being practiced by many communities in India. We took large number of samples, 275 population groups, 2,800 samples, and try to understand how two unrelated individuals share a common DNA stretch. We call it as haplotype. The reason is, if there is a DNA change, we call it as a mutation in the forefather, and if the mutation is called as recessive mutation, which would cause recessive disease in the sense, if the mutation is in one of the chromosome, it will not cause any phenotypic change of the individual.
Right. So the person remains healthy.
Healthy.
But is like a carrier.
It's
right?
a carrier. You can call it as carrier. If two such individual, boy and girl, carry a mutation in one of the chromosome, if they marry, there's a chance of 25% chance that they contribute the abnormal chromosome to the next generation. Therefore, there's a disease.
Right.
With this idea, if you look at we scientifically called as IBD score, identity by descent. If two unrelated individuals share a common haplotype or DNA stretch is identical, if that carry a mutation, if the two unrelated individuals today, that means they are from the same common ancestor, thinking that they are not related, and if they marry, that's when the problem is going to start.
Okay.
Such a condition is very, very high among Indians. At least 1/3 of the population which we analyzed out of 275, 81 of them have high IBD score. That means the identity by descent score is very, very high. That means although today they are unrelated, but several generations back they are related. So the founder has the mutation, or forefather or foremother has a mutation. It would have circulated in this population, and the frequency of that mutation must have increased. And two of them who carry the mutation in heterozygous condition, that means one chromosome is normal, one is affected, there's a chance that they marry and give rise to individual with the disease.
Okay.
So that condition is high among Indians. If you compare to Ashkenazi Jewish population and Finnish population, these are the two populations has been well studied for endogamy and the impact of endogamy, and large number of disease has been reported, and large number of mutation which are associated with the disease also been reported. So it's very well known fact. So we compared these two populations with large number of Indians. We found large number of Indians are several-fold higher chance of having mutation and the disease compared to Ashkenazi Jewish population and Finnish population. That's very, very dangerous. So each one is even worse than that. If you talk about the disease pattern. So large number of them. Although today we don't know how many of the populations are carrying the population specific disease, but the genetic is reflecting that yes, definitely there is a large number of population specific disease, and most of them are not even come to the attention of the medical community. It's very, very sad.
Could you maybe give us a few examples of this, the kinds of communities that you've studied over the last several years, and the kinds of diseases with strong genetic links? Now, from what I understand, there are many conditions from diabetes to many cholesterol-related illnesses, which are very complex, right? I mean, they have a genetic component, they have environmental component, our health history, and all of this. But there must be some which are pure genetic diseases, which I guess are easier to study.
Some disease, like the multifactorial disease like diabetes or cardiovascular disease, there are several genes are involved in causing the disease, in addition to the environmental factors, our lifestyle, and many more factors. And also these diseases are common. So if you take 100 individuals, you will find at least 10 of them are diabetic, or 5% of them are cardiovascular disease, and many more. So those are very common diseases. So what we are talking is the rare and the population specific disease. That means the disease is restricted to only that population.
Right.
So that kind of thing, you find 1/3 of the population you analyze are likely to have such kind of diseases.
Dr. Thangaraj, so we started off with having two populations, the ancestral South Indian and the ancestral North Indian, who have admixed in various ways to create these different subpopulations that have now stayed endogamous for the past 2,000 years. Do we have any understanding of when or how they might have picked up these unique mutations that are causing these diseases to be present in one subpopulation but not in the other, while they are in the same geography, right? So they're all within India, yet we have different mutations that have turned up.
One can predict the age of the mutation if we identify the mutation. For example, we identified a mutation which causes, this is called cardiomyopathy. There's a 25 base pair deletion in a gene called myosin binding protein C3 gene. So we identified that mutation, then we sequenced the entire gene of human and also entire gene of chimpanzee, and based on the mutation rate, we predicted that was probably arose 33,000 years back.
Okay.
Similarly, if we identify a mutation responsible for disease, we can be able to understand the age of the mutation. But before that, we actually have to identify the mutation which is responsible for the disease. So that's a big challenge.
So tell us a little more about cardiomyopathy from this particular source. What did you find in Indian populations?
The cardiomyopathy is one of the heart muscle disease which leads to sudden cardiac death. And there are three different kinds of cardiomyopathy. One is a dilated cardiomyopathy, where the ventricle is enlarged beyond the normal size. Other one is hypertrophic cardiomyopathy. The heart muscle become very thick. And the third one is restrictive cardiomyopathy, where the heart muscle become very stiff.
Okay.
So because of these conditions, the efficiency of the heart for pumping the blood goes down, suddenly it stops functioning, therefore sudden cardiac arrest, people collapse. We analyzed large number of individuals, particularly one of the mutation which we found present in the myosin binding protein C3 gene. There's a 25 base pair deletion, which actually removes one of the exon, that's a coding region, protein making region. Therefore, the protein is truncated, small size protein, therefore it's unable to do its function.
Right.
Hence, it leads to this kind of phenotype and the disease. When we look for this mutation in Indian subcontinent, we found on average about 4% of them are carrying this mutation.
Okay.
In a heterozygous condition. In a heterozygous condition, earlier I mentioned that one chromosome is normal, one is with the mutation.
Right.
When the individual carry the mutation in heterozygous condition, they live up to 40 or 45 years without any symptoms. But after 45 years, suddenly either they're running or playing or cycling, they may collapse, and on the spot they may have to lose their life. But in the mutation exists in the homozygous condition, that means both the copy of the chromosome carry the mutation, the individual die at very, very early age, less than 10 years. That we have shown by looking at the postmortem tissue of the children who died because of the sudden cardiac arrest. So we found a large number of them carrying the mutation in a homozygous condition.
Okay.
When we try to look for its distribution all over the India, as I mentioned earlier, it is existing almost in every state at 4.5%. Then we try to look for how it is distributed all over the world, and to our surprise, we found that this mutation is restricted to only to South Asian population, but not in the other countries or on the other continent, suggesting that Indians and South Asians have very unique set of mutation, which is not present in any other country. This is one example. Similarly, there are several examples for many diseases, suggesting that particularly if you take cardiovascular disease, the frequency of individual with the cardiovascular disease were quite high in India. So this could be the reason why we are more prone for cardiovascular diseases. Similarly, there are many examples.
there's research from, say, the United States about how the African American population in the US might suffer from a higher rate of diabetes. So similarly, any examples of how individual communities in India have a higher predilection for one disease or another?
One of the example could be the Vaishyas. So Vaishyas are a small community in Andhra Pradesh. Now they are also in Telangana. So they are, for the last several years, following a very strict endogamy. And several years ago, it has been found that they have a problem of pseudocholinase enzyme deficiency. So because of that, when somebody from Vaishya goes to hospital for surgery, if the regular anesthesia is given to them, most often they may not come out of the anesthesia because of certain unique mutation which has not been found so far. Every drug which we take, there are genes which metabolize, breaks down. Therefore, we will come out of the anesthesia after some time.
Right.
But here, because of the mutation, they are unable to metabolize the drug, and therefore they are in the impact of the drug for very, very long period and may lead to paralysis.
Okay.
So although the gene has not been identified yet, or the mutation has not been identified yet, but what we found was for the last 108 generation, this particular population is following the endogamy marriage.
Okay.
So there is no gene flow on both the sides. That means that they are not marrying anybody from outside and bring them into their community. The same way, nobody is going out of this community and marrying outside. Otherwise, this condition would have changed.
Okay.
So this is one example. There are several examples where you can see that there's a regional specific diseases. For example, Handikudu disease. There's a place called Handikudu in Karnataka. In that place, most of them are with disease where their bone, both the hands and leg bones are bent, and mostly they are in short stature. Because very unique condition. That's why it's named after the village as a Handikudu disease. That's again that regional specific. Probably some founding mutation which makes everybody in the village or most of them in the village are with this particular disease. So there are several examples one can...
and I'm sure there are thousands more that need to be researched.
Yes. So these are the one which is just come out now. There are many more. For example, some diseases, some mutation which may lead to very early death, premature death. There's no way that information is reaching to medical community or any health officials. In the village setup, if somebody dies one or two, they're not going to report anywhere. So like that, I'm sure that there could be many, many more conditions in Indian subcontinent and also other South Asian countries, because India contributed about 50% of the genetic material to our nearby South Asian countries. So the condition, what we see in India, is not only restricted to India, so we also expect that in South Asian countries.
And by South Asia, you mean not just Pakistan, Nepal and Sri Lanka, but also maybe Indonesia, right? Because we have a population...
those who are migrated recently from India to other countries, Southeast Asian countries like Indonesia, one can also see that. There are some conditions where Indians settled in London for several generations.
Right.
Have been found with a specific mutation causing to neuromuscular disease. And in their health record and the database, they didn't find this mutation. When they try to, when I collaborate with them, they try to understand the origin of this mutation. So they asked me to look into the patients from our collections and found, of course, we didn't find any of those individuals, any of the individual who have a similar disease carrying that mutation. That could be some very unique mutation restricted to that population. So we don't have the population information to go back and check it. On the other hand, there's also one example where in Europe, they have found a mutation which is responsible for microcephaly.
Okay.
Okay, so they
Shrunk
are Indian
heads.
origin. They try to understand about the origin of that mutation. Basically, they are from Sri Lanka, so we don't have Sri Lankan sample. So they try to understand whether that mutation is existing here. We found in our populations 3 out of some few thousand individuals carrying that mutation. So, of course, we didn't see the impact of that, but similar mutation we also found in South India. So, it's not only useful to India and also useful to Indians who are living in other countries.
So, Dr. Thangaraj, if the Finnish population and the Ashkenazi Jew population, with the Jewish population, which is one, I guess both are more financially well off than most of our communities in India, and the problems have been better studied. Are there any systems that they have set up to help manage these kinds of problems where, say, there is a risk of two people getting married and passing on a recessive gene to a child? Is there any way to prevent it?
Particularly the Ashkenazi Jewish. So they have an organization called Dor Eshrim. And this organization, for the last several years, so they have made a database of the children. So they go to school, collect the samples, blood samples from the children, sequence them, put it in their database. And when the children grow and they come to the stage, the boy and girl want to marry, and they go to the organization, Doresh room, and identify that boy, identify a girl, this is the girl I want to marry. Then actually they check, go back to the database, check their DNA sequence, whether both of them carry a same mutation. Because the system is well established, they sequence all the disease condition, they identify the mutation. So they can just go back and look whether both boy and girl having the same mutation. If they have the mutation, they advise not to marry first. Even they wish to marry, they say that, okay, this is the problem, you have this mutation, both of them, in heterozygous condition. They explain that what would be the consequences if you marry. There's a chance of 25% having children with the disease. So you better use the prenatal diagnostic test every time and see whether the mutation exists in the homozygous condition, the fetus, or not. If there's a homozygous mutation, absolutely true that they're going to have a child with the disease. If the heterozygous condition, no problem. It's a carrier like both the parents, so there will not be any problem. So they can continue. So that way, one can actually predict the health condition or actually give a better future generation without the disease. So in this way, the Ashkenazi Jewish populations have eliminated a couple of diseases from the population itself.
Wow.
So by looking at carefully with the counseling and so on. So that is the one which actually need a country like India, where we have seen that most of the Indians are having several-fold higher chance than the Ashkenazi Jewish population to have a population-specific disease.
In the Indian context, Dr. Thangaraj, would you advise this to be done at a population level? So the 5,000 odd subpopulations that we have,
Yes.
They should do this individually for their own communities.
Yeah, it would be better to do all the populations, and if not all the population, at least the population where it would be a good idea to begin with the population where we identified most of the population which has a high BD score. So that's the first step. Then go to the next step to expand the study to cover almost all the population. So of course there's a cost involvement is very high in this, but it's worth spending now to avoid future generation with so many diseases.
I like what you said that, usually we talk about healthcare or we talk about preventive health. But what you're talking to us about is actually about predictive health, right? We can actually anticipate what might come, take corrective measures, or at least keep our eyes open for something that is upcoming.
Yeah, it's both predictive as well as a preventive medicine. So you first predict, yes, you have mutation which may leads to the disease, then it's going to prevent. So predict, then prevent, and give healthy generation to the future.
So just a couple of questions at the end, Dr. Thangaraj. You said that this will cost a lot of money. Could you just give us a sense of how much doing a full genome sequencing costs today?
So full genome would cost few lakhs rupees.
Okay.
But rather than going to full genome for everybody, we can actually target only the exomes, because the exomes are the one which is coding region, responsible for making protein, which is essential for all the function. So we can actually target only all the coding regions, which may cost around 25,000 to 30,000 rupees per individual.
Okay.
Then we don't have to sequence all the individuals of the same population. You can select initially based on the phenotype, few families. Maybe let's talk about 100 individuals who are with the disease and their parents. Once we sequence all these 100 individuals, identify the mutation, so that can be screened in all the population. Right. So once they have the data in hand, then we can actually counsel them whom they should marry, whom they should not marry, or even if they are very strong that they don't want to follow the non-endogamy practice, yes, okay, you can follow, but one can do the prenatal diagnosis to avoid the fetus with homozygous mutation. So that way, if you practice this for few generation, you can actually end up in getting the very healthy population in the future. Today you may be investing a huge amount of money, but it's all will save what we are going to spending in treating the patients. Rather than treating the abnormal patient, spend money to avoid all complications to have a healthy generation.
Thank you so much, Dr. Thangaraj. As it's good that we are having this conversation in 2018, because this is also a time where I think health has become a political subject for perhaps the first time in Indian history. Individual famines, individual diseases were always captured public imagination.
Yes.
But the idea that now we are talking about, be it Ayushman Bharat, or universal insurance or something, to me it is a sign that health is a subject whose time has come in India.
Yes, I agree.
And, bold new policies need to be formulated.
Absolutely.
And here's to hoping that we understand India's population and its genetic diversity well, so that we can manage our healthcare much better.
Yeah, thank you very much. That's a great view to have something of this sort, which would be even more better if this reaches to the public and reaches to the government, so that some positive information comes and would be helpful for the future generation.
Dr. Thangaraj, it has been great having you here, and your research clearly underscores all of this predictive medicine that going forward would really be beneficial to the country. I wanted to ask how many other research groups in India or worldwide actually do this kind of research focused on the Indian population?
For the Indian population study, not many. Professor Partha Majumdar's group in Calcutta does it. But linking both the population and the health is not in many places.
I guess some groups work on the genetic links to disease.
Yes.
And maybe some others work on just populations and public health.
Yes.
Right. So, but this is bridging the two.
Yes, bridging the two.
So we clearly need more research that kind of bridges this. to be able to get a clearer picture of what India needs going forward.
Right.
So on a personal note then, Dr. Thangaraj, how did you get interested in doing this kind of research?
Yeah, we started about 20 years back. Actually, during my MPhil, I did some population study. PhD, of course, it's entirely different cytogenetic aspects of mental retardation. So I've been doing genetic study right from my MPhil and PhD days. And when I joined CCMB, there's an opportunity. I joined with Dr. Lalji Singh, who initiated some of the population study. So at that time onwards, so we initiated and we established large number of collaboration within India with several universities and colleges where students used to come and do the dissertation work, who bring the sample from their own place. Otherwise, collecting such a huge number of samples is very difficult. So each one used to come with 100 samples from different community. So do the work, and we used to publish the study along with the students. Then the DNA remains a part of CCMB. That's how we have huge collections of the samples.
Dr. Thangaraj, thank you again for coming on the show. It was an absolute pleasure to have you, and it was an absolute pleasure to talk to you about everything from human history to modern healthcare challenges.
Yeah,
Thank
you very much,
you.
and it's my pleasure to be here.
Thank you for staying with us till the end. If you have any questions for Dr. Thangaraj or for us, do write in to podcast@thinkpragati.com. Visit our website at thinkpragati.com for your daily dose of brain fodder on all things public policy.
You can subscribe to the Pragati Podcast on the IVM Podcast app or wherever else you get your podcasts from. We're there everywhere.
Look! Up in the internet! It's a meme! No! It's a cat video! No! It's the Geek Fruit Podcast!
That's right, we interrupt this riveting broadcast to tell you about our show, the Geek Fruit Podcast, where Tejas, Dinkar and I, Jishnu, talk about everything in pop culture, including DC, Marvel, Star Wars, Netflix, and everything in between.
You know how your friends hate it when you ramble about some nerdy crap and you just want somebody to listen to you? Well, sorry, there's nothing we can do about that, but come listen to us ramble and it'll almost be like the real thing. Kind of.
Listen to new episodes of the Geek Fruit Podcast every Monday and the Geek Fruit Bulletin every Thursday on iTunes, Google Podcasts, the IVM app, or wherever you listen to podcasts. Happy listening, you nerds.
Did you know that Parsis in Mumbai, instead of being left at the Tower of Silence after they die, are now cremated? And why? Because a cow fell sick in the early 1990s. Did you know that the smog in Delhi is caused by something that farmers in Punjab do and that there's no way to stop them? Did you know that there wasn't one gas tragedy in Bhopal, but 3? One of them was seen, but 2 were unseen. Did you know that many well-intentioned government policies hurt the people they're supposed to help? Why was demonetization a bad idea? How should GST have been implemented? Why are all our politicians so corrupt when not all of them are bad people? I'm Amit Verma, and in my weekly podcast, The Seen and the Unseen, I take a shot at answering all these questions and many more. I aim to go beyond the seen and show you the unseen effects of public policy and private action. I speak to experts on economics, political philosophy, cognitive neuroscience, and constitutional law so that their insights can blow not only my mind, but also yours. The Seen and the Unseen releases every Monday. So do check out the archives and follow the show at seenunseen.in. You can also subscribe to The Seen and the Unseen on whatever podcast app you happen to prefer.